Well, thanks everyone for joining from many different time zones. What I'm very excited to talk to everyone about today is a project that we initiated as a novel therapeutic strategy for NEB-related Nemaline myopathy. And this is a project that has its origins of funding from AFBS and has now led to additional broader funding, which is very exciting and hopefully enabling us to move this pretty quickly with the goal, obviously, of hopefully bringing this to patients. I don't think I need to go over this background, but I think I would probably highlight three things. One is the idea of a common underlying mechanism for Nemaline myopathy related to the thin filament. And then I think two important issues. One is that while there've been a lot of fundamental work understanding the thin filament and the nature of mutations that cause Nemaline myopathy, few candidate therapeutics have emerged. And secondly, we're still kind of in the early stages in defining disease natural history, and most specifically identifying outcome measures that would be suitable for interventional clinical trials. And I highlight that because the way that I think about drug development is really a two parallel pathway. A lot of emphasis is always placed on finding the therapeutic, but I think what we've learned from some of the other, I must be on some timer thing, so apologies. What we've learned from some other muscle diseases, and in particular, some of the field's work with another congenital myopathy called X-linked myotubular myopathy, was that the clinical trial readiness piece is equally as important, and making sure that when there is a candidate therapeutic, that there's also everything needed to bring that to patients at that time. And I'll talk a little bit about what's happened so far and where I see the future for that as well. So I'm going to specifically focus the therapy development part on Nemaline-related Nemaline myopathy, though we do have some ongoing studies related to other forms of Nemaline myopathy and therapy development. I think probably everyone in the audience is familiar with Nemaline, which is one of the largest genes and encodes one of the largest proteins in the genome. And Nemaline's function is in the thin filament, and its primary role is to establish the length of the thin filament, which is directly related to force generation. And reduction in force generation is directly proportional to muscle weakness. So Nemaline obviously has a fundamental role. It's a very large gene and therefore not amenable to classical gene replacement therapy when one thinks about a genetic strategy. So there are good models, animal and cell models of NEB-related Nemaline myopathy. Several years ago, and again, with support from Foundation Building Strength, we generated and characterized a zebrafish model of Nemaline myopathy due to NEB mutation. We've now characterized, I think, up to 10 different Nemaline mutation models, and hopefully soon we'll be reporting on our findings with mutations all across the gene and how they may differentially affect the function. Cohen Ottenheim and Henk Grenzier, who you may be familiar with, generated a mouse model of the most common mutation in Nemaline, the exon 55 deletion, several years ago. And this model has been used for mechanism studies, but has proven to be a challenge from a therapeutic perspective because of the severity of the phenotype of these mice. And this is something that we've made some new insights into and think we have a strategy for potentially making them less severe. And that's part of the current ongoing project as well. So we first started with a question of whether or not we could use a genetic strategy to find things that modified the phenotypes of these animals as a conduit for identifying new therapies. We put a lot of work into two different approaches. One was a drug screen in the Nemaline zebrafish. And again, this was supported initially by Foundation Building Strength. And secondly, we performed a genetic modifier screen in the mouse model of exon 55 deletion. And frustratingly, despite pretty intensive studies, we were unable to identify any modifiers. And while I don't think this means that modifiers don't exist, what it told us is that this strategy may be challenging. And also the idea of using second site, say, small molecules that are drugs that might modify pathways, may be a challenging approach for therapy development for Nemaline. And therefore, we reasoned that maybe gene-based strategies are really going to be the way to go for this. Again, I mentioned that the gene is too big for conventional gene therapy. So we thought that perhaps a different strategy, in this case, CRISPR gene editing, might be the right way to go. Just to give a little bit of background on CRISPR, people have probably heard about this. This has revolutionized science in the past few years, including a Nobel Prize being given to the founders of the idea of this. CRISPR stands for clustered regularly interspaced short palindromic repeats, which is obviously a mouthful, which is why we use CRISPR. And this is a bacterial system. It sounds kind of funny, but bacteria actually have their own immune system. And this is how bacteria fight off viruses. They basically use the CRISPR system to inactivate viruses. It has two core components. One is an enzyme called a nuclease, which can cut DNA. The one in bacteria is called Cas9, and now there's been several of these recognized across different types of bacterial species. And the other is the molecule that guides the Cas9 to the specific sequence for it to cut, and that's called a guide RNA. The way that it works in bacteria is the bacteria gets infected with a virus. It then integrates part of the viral DNA sequence and uses that to create the guide sequence, and then heads back to the virus with that guide sequence to target the Cas9 enzyme to cut up the viral DNA and therefore inactivate it and prevent it from replicating and reproducing and going off to infect other cells. So this idea has been co-opted into mammalian cells for many different strategies. Really, the way that CRISPR has been most successfully used so far is to create new mutations. So over the past couple of years, there's been a total shift in how we make animal models from the sort of pre-CRISPR way of doing it to now really making most of our mutations in zebrafish or mice or whatever species using CRISPR-Cas9. And that's because it's very effective at targeting very specific mutations. One of the other things that can be done is you can use CRISPR to repair mutations. And the way that you can do that is by providing not only that guide RNA, which can target the cutting to a very specific place, but you can provide a template that basically has normal sequence that the cell will then use as a basis for repairing. So the Cas9 goes in and cuts and makes basically a cut in the DNA, and then the cell tries to repair it. And normally, without any guide, it will repair it somewhat inefficiently, and it will cause either deletions or sometimes insertions of sequence, but very rarely the exact normal sequence. But you can provide a template that will steer the cell to repair it correctly. And we thought we could potentially use this strategy for Nemaline myopathy. Basically, the idea of using a guide to a specific sequence in the Nemaline gene, and then a repair template that could then fix whatever mutation was at play. And we decided to target the most common mutation in Nemaline, which is the exon 55 deletion, which many of you may be familiar with. This is the common whole exon deletion that was first described in Ashkenazi Jews, but has really now been seen to be really a mutation that impacts individuals around the world. And I think remains the most common Nemaline mutation. And this is a large deletion that includes 2.5 kilobases of genetic material and includes one specific exon, this exon 55. And when this is removed in patients, what happens is the RNA is still made, but the protein is not stable and gets degraded. So patients with exon 55 deletion have diminished amounts of Nemaline protein, which then leads to Nemaline myopathy. And so our idea was to use a guide RNA targeted to the remaining intron sequence between the exon 54 and 56, the Cas9 enzyme to make a cut, and then a repair construct that includes the entire exon 55, which hopefully then the cell would use to re-add in exon 55. And at start, I have to say this was a bit of I won't say far-fetched, but something that no one had really necessarily tried before, the idea of having such a large repair. And so we weren't sure if it would work. And we tested this first out in cells as a means of trying to see, just as a proof of concept. And this was our basic strategy. We have a bank of fibroblasts, so skin cells that were created from skin biopsies of patients with exon 55 deletion Nemaline myopathy. And we grew these cells in culture, and the plan was to add the Cas9, the enzyme that cuts, the guide RNA, and then again our repair template into the cells, use a little trick to convert the skin cells to muscle cells, and then grow them up and look at the Nemaline expression. This is just to show what the cells look like. On the left is the fibroblast, the skin cells, and on the right is our little trick to turn them into myotubes, which is a mature form of muscle cell. And what we can see is when we do this in normal, in non-Nemaline cells, we get good expression of Nemaline. And then in the patient cells, we see an absence of the Nemaline protein. So consistent with the exon 55 deletion phenotype in patients. So then we used our strategy of taking the Cas9 guide and replacement, put them into the cells, grew them into myotubes. And what we were able to see, and then this is in a cell line from an individual with the exon 55 deletion, that we could successfully reintroduce the exon 55 into the DNA and then also into the RNA, suggesting that this repair not only can happen, but actually the new exon that we give can be incorporated into the RNA that's made from the DNA. Of course, this is not fully convincing unless we could see that the Nemaline protein is retained. And we were able to see this. And so this is now using a technique called immunofluorescence. And on the left is two myotubes from a patient exon 55 deletion cell. Red is alpha actinin, which is just marking the Z band, which is the part where the thin filament anchors. And then in green would be Nemaline staining. But as I mentioned, these cells don't express any Nemaline protein. But then you can see on the right is a myotube from the same patient cell line, but now given the Cas9 guide RNA and repair template. And you can see because it's now got green and red, it's turning yellow, meaning that now we've got re-expression of the Nemaline protein. So successful reintroduction of exon 55 leading to re-expression of the Nemaline protein. This is a different patient cell line showing the same effect. So here's here's the patient cell without getting the CRISPR-Cas9, and this is one getting the CRISPR-Cas9. And I think you can see two things with this one. One is that not only do we get re-expression, but also the myofibers look more robust because we think we're restoring the thin filament length in them. Though this is a bit hard to control in the patient cells because of the nature of this strategy of using this skin cell, turning them into myotubes. They're very immature myotubes. So all this is to show that we could really, that the technique works, that we can use CRISPR-Cas9 to make a cut in the DNA and then use a repair to reintroduce the exon 55. So now what we want to do is see if we can use this strategy to see if it can rescue the phenotype of an animal model, because we think this will be essential to see in order to consider potentially moving this to the clinic. And again, we're using the same strategy, the Cas9, the guide RNA, and the targeting template. And what we're doing is we're packaging into a viral vector called AAV9. And AAV9 has been very commonly used in other gene therapy programs, including the recently approved spinal muscular atrophy gene therapy program, which also uses AAV9. So we know that it's safe in patients and very effectively delivers to all the cells in the body, including the muscle. And we've been trying now various strategies to introduce this construct into the mice as a way of trying to see if we can A, create the same rescue of the exon 55, and B, to see if that actually improves the phenotype in the animals. And so here's where we are to date. We've been able to show both with intramuscular and intravenous infusion that we can achieve the cutting, and also that we're getting some low levels of repair. We're now working on optimizing the efficiency because our cut repair amount is about 20% cutting and about 1% repair, and we think we're going to need more of that. So we're optimizing the amount we're giving and the sequence strategy. And then the other thing we're doing is trying to work with that mouse model that I said has a very severe phenotype to try to A, make it have a less severe phenotype so it's more workable from a drug development strategy, And the other thing is that we're creating a part in that mouse that has human sequence. And that's critical because, ultimately we need to develop these reagents for patients. If we use a sequence, it's that guide that targets the Cas9 only to the mouse sequence, that's not going to be an effective reagent. We'll have to then remake that for human cells. And the good news is we already have the human cell guide sequence because we've done it in the patient cells. So we're creating that landing pad in the mouse. So Mark, do you have a question or?
Well, just I wanted to clarify a little bit. So
yeah,
I mean, I've heard this many times, so I wanted to make sure it was explained well. Like, so the current, excuse me, the current mouse model that had been developed by Hank Granzier's group is very fragile, meaning that it was not living long enough to actually test therapeutics on it, but they were able to use the muscle cells that actually seemed to look like under a microscope like the muscle cells of the humans. They were able to test on it, but not able to utilize the living mouse, because the living mouse, the mouse was living for maybe 9 days. And so it wasn't long enough. It was too fragile. And so that's when he was saying the phenotype was, too vulnerable. That's the expression, is that the mouse cells are working and presenting like the human cell form. And so they were able to use it for experimentation, but they weren't able to use the actual living mouse for therapeutic models. That's why I just wanted to go into that just a little bit more deep, because the phenotype thing can be hard to understand.
Yeah, thanks for clarifying. And it turns out actually that the mutation in the mouse we've just discovered creates a different change than in patients, that we think is why the phenotype is so severe, why they're not surviving very long. And so part of the changing and making it with the human sequence there will also hopefully remove that change that we're seeing and make them live longer. So just to give an example, in our hands, these mice live about 5 days. And when you put AAV in, it takes about 2 weeks to get maximum expression of whatever you're adding. So you can imagine if we're adding something in at the first, right after they're born and they're dying 4 days later, we're not going to get hardly any expression of the repair part. And so it becomes kind of a very challenging thing. So we're trying to actually give it when the animals are inside the mom, because that gives us a longer window, but that's also not necessarily as translatable to patients as it would be if we give it after we see the disease has developed. So that's why we're doing the change. But thanks for the clarification. I see there's a hand up. Yeah.
Yes. So question, I didn't really follow. So the mouse model, it doesn't live long enough, so you can see if it actually works. That's right.
Yes.
And do you have any mouse models on developing, I don't know, where they will live long enough to be able to test on?
I think that's what he was saying, right?
Yeah. So we're making that now.
Okay.
Yeah. And the other thing we can do that we are doing is we can use the mice that have only one that have a mutation only in one copy of the Nemaline that make that are what are called heterozygote, basically the carrier mice, and we can do some testing on those mice to at least perfect the efficiency of the targeting, because they'll have one of the two copies that's got the mutation, so we can do the repair there as well. The challenge again has to do a little bit, it's a little bit, I wasn't going to get into this too much, but the logistics of what makes the mouse not suitable has to do with the fact that, so in patients with exon 55 deletion, so there's you have DNA that gets transcribed or made into RNA, and the RNA gets made into protein. And so the exon 55 deletion in patients makes a normal amount of Nemaline RNA. That RNA gets made into a protein that's missing the sequence of exon 55. And for reasons that are not totally clear, the missing exon 55 sequence makes the protein not last very long, not stable. And so it gets degraded and you don't have very much protein, but the RNA levels are normal. in this mouse model, even though it's the same mutation made as in patients, not only is the protein missing, but the RNA is missing. And that's because when the exon 55 deletion is made in the mouse, it's creating a second change that was unanticipated, that up until now had not been understood. But we used a different kind of molecular approach that found out why that was happening. And now that we know, we can actually correct that second problem so that it's just should hopefully behave just like the muscle cells do in the patients. I know it's a bit, it's even it's complicated from a science perspective. So I'm trying, it's a little bit hard to necessarily communicate, but I think the upshot is that we need to make the mice have some challenges that make it not perfectly suitable to test our approach, but we're confident that we have a strategy to overcome that so that we can use the mouse as a way of testing this. And there are other workarounds if the mouse doesn't ultimately work out, but I think we'd really like to see this strategy effective in mice, because that gives us a sense for how much repair is needed. So one of the challenges I can talk about with the CRISPR strategy is the efficiency piece. So even the most efficient, so there's been a lot of work done for a different muscle disease called Duchenne muscular dystrophy to optimize the CRISPR strategy. And fortunately, my colleague whose lab is right next door to mine is one of the world experts in developing CRISPR for Duchenne muscular dystrophy. So we're learning a lot from him about the best way to do it. So he's done all the troubleshooting. We're taking from him the most optimal parts and applying it to Nemaline myopathy. And so we're not sure how much replacement and repair we'll get. And so one of the key questions we'll want to ask is, how much do you need to get the muscles to function again properly? Now, you could extrapolate from the fact that individuals who are carriers of the exon 55 deletion don't have muscle weakness to say, well, 50% of the normal amount of Nebulin should be enough, but we might not achieve 50%. And my suspicion is that less than 50% will still be fine, but we just need to find out what that is. And one of the things that we're working on is to define how much is going to be needed, because that will really predict whether or not this strategy would be successful. So I had a little bit on the natural history study, which I can do very quickly. Just to summarize. So our hypothesis again is that we can use this gene editing approach to reintroduce exon 55 into the Nebulin locus to then restore the normal RNA and protein, and therefore provide a therapeutic for the condition. We were successful in showing it in patient cells, and now we want to do it in an animal model so that we can really reinforce the effectiveness of it and define the characteristics that will be needed to successfully bring it to patients. And I also wanted to mention that we are working on other gene-based strategies for Nebulin as well, that are not only for exon 55, but for other mutations, and also for ACTA1. And I think there's some good considerations of gene-based strategies for some of the other forms of Nemaline myopathy as well. So for example, KLHL40, KLHL41, and LMOD3 are all of a size that they could fit into a typical gene therapy vector. So they're good candidates for gene replacement therapy, something that hasn't yet been done, but I think is a potentially effective strategy as there's more and more knowledge on how we can do gene therapy generally for muscle disease. So just quickly to talk about the natural history study. If you haven't had a chance to see it, so we recently did publish this study in 2021, which we called a cross-sectional study of Nemaline myopathy. So this was a project supported by Foundation Building Strength that came out of the two family meetings that occurred about 6 years apart, so 2009 and 2015, where participants very generously agreed to have neurological exams and also to do clinical histories and participate in some outcome measure studies with the goal of seeing get some understanding of the condition in general, but also really to start looking into that question of what might be suitable things to look at in an interventional clinical trial. So this is just the general characteristics. There were 57 individuals who participated in the study, 16 of whom were both in 2009 and 2016. Pretty broad age range. And unsurprisingly, in terms of a breakdown of subtype, the majority of individuals had either ACTA1 variants or Nemaline variants. And also, I think not unsurprisingly, in terms of the sort of Monique Ryan, Kathy North clinical criteria, had typical congenital Nemaline myopathy. And so I just wanted to emphasize a few things. One is that we were able to successfully show that some of the outcome measures that have been used in other muscle diseases seem very suitable for Nemaline myopathy. And here, if we focus just on the bottom panel, this is a scale called the MFM, or motor function measure, which has been the primary outcome measure in several different clinical trials for other muscle diseases. And it seems to be a very good score for Nemaline myopathy, because it is that, with these types of scales, the importance is seeing whether or not everyone is either kind of at the floor, at the ceiling, or in the middle. And the good news is that the majority of patients with Nemaline myopathy are not at the ceiling or at the floor, but somewhere in the middle. And in particular, one of the domains seems to be most sensitive to detecting changes in the condition. There's also some of the timed outcome measures. It seems like they would work. So for example, the 4-stair climb and the 10-meter walk-run test, which we were able to do in several of the participants. We also were able to see that looking at lung function through pulmonary function testing seemed to show changes in the majority of individuals, and also would be suitable as a potential outcome measure. And finally, we looked at a novel outcome measure called the Slurp Test, which is something developed by Tom Crawford at Johns Hopkins University. It's a very simple measure where basically take a cup of water and an individual has to drink through a straw. And I think also, not that surprisingly, everyone who participated in the study had an abnormal slurp test compared to normal values. So I think this could be a nice way of measuring the dysfunction that occurs in many Nemaline individuals in terms of what we call bulbar function, so chewing and swallowing. And this was just the longitudinal component. So again, there weren't that many. We only had 16 individuals that were at both time points. So this is, again, quite limited. But the upshot was that we didn't see a lot of change in the individuals who participated in 2009 and 2016. So the disease course, at least in that small sample size, seemed quite stable, which I think has been the general experience with the condition as well, particularly after the first year or two of life. So just to summarize, we believe we identified some outcome measures that would be suitable for use in clinical trials, in particular the MFM32 and pulmonary function testing. We also think this novel outcome measure, the Slurp test, could be one, particularly if we could work a little bit to make it more uniform between individuals. And then we had our small longitudinal component that showed disease stability for 16 individuals. I think this highlighted the fact that clinical trials are definitely going to be feasible for individuals with Nemaline myopathy, but likely we will need a dedicated longitudinal prospective study to really define and refine what those measures are going to be. My cat wants to say hello. To best prepare for when therapy is available to test. And I just wanted to mention one more thing, and I'm mentioning this with two hats. So we have a recently formed congenital myopathy task force that's being sponsored and developed through an international organization called Treat NMD. Treat NMD has been around for a while. It originally started as a European Union funded initiative, but now it's an independent initiative. And historically, it's really focused on DMD, Duchenne muscular dystrophy, and spinal muscular atrophy. But one of the goals now is really to have it as a vehicle to help with other conditions where there has been traditionally less attention. And congenital myopathies in general, and Nemaline in particular, is a condition that's very amenable to the approaches that Treat NMD has used historically. And in that vein, we've actually established a congenital myopathy task force to hopefully, with the goal of addressing some of these gaps in current clinical care and also in clinical trial readiness and therapy advancement. And it's really supposed to span all those areas and try to help fill in gaps where they're needed. And this is the leadership team, and you'll see some names you probably recognize as being people who've been involved in Nemaline myopathy over many years. And one thing I wanted to highlight is we're hoping eventually to have somebody on the leadership team who's from family advocacy groups. And our thinking is to start by having basically a town hall, inviting all individuals who are part of family advocacy groups to come together to discuss what the feeling is for the needs of the field, who should be the people discussing it, and also potentially who might be a good representative or two from that group of individuals. And so, all of the individuals on today's call, you're certainly welcome to join in on this. And when that comes, I can certainly pass on the information to say what's going to happen. And I should mention that I'm leading this task force, and I'm also currently in the position of chair of Treat NMD, which I view as a win for congenital myopathies, because again, it's not a condition that's historically been addressed by Treat NMD, but we're really hoping that that's going to change now.
Jim, so just to add to that point,
oh, yeah, sorry.
So our foundation received the PCORI grant this year, and so that's a very patient-centered approach. And so what they've encouraged us to do, and part of our appeal for the grant indicated that we would bring patients and researchers together in group meetings 2 times over the next 2-year period, and that we would also have 10 different webinars set up where there was an advocacy group, like a patient slash Nemaline focused advocacy group with researchers, so that the researchers are really being influenced in a positive way by the community. And so that's the primary focus. And that was a $250,000 grant that we received that spans over a 2-year period with very, very specific deliverables. And so that's a work that we're going to be undertaking. I think the grant starts in January. So we will be communicating to this group here. And if there's any way to sort of interface and interloop the two, that would be extremely beneficial.
Yeah, that's great, Mark. I think what one potential, so that I think is a good thing to potentially think I can to bring up in the town hall, but also one thing that's going to be happening in early 2022 is an educational masterclass on congenital myopathies, geared really towards clinicians and care providers. But that might be an opportunity as well to have the PCORI grant introduced to that group and how and talk about where that interface can occur moving forward. So I can definitely provide more information related to that.
Great. I just want to go back and thank Boris and Sharon Shimanovsky for really being the key drivers in getting the Greenwald Foundation to contribute $250,000 to get this, the project with the CRISPR-Cas9 work on the Nemaline gene off the ground. I think it was a great, win for the foundation to really continue the momentum that Jim's already done in his lab and really take this to the next level with a 1-year specific time frame project, so that we'd really try and push the envelope and see results within that time frame. We love looking at long-term studies also, but really want to see what kind of change we can make in shorter term periods that would affect our patients. So thank you. I wanted to give you a chance to do through this slide too. Sorry.
Yeah, no, thanks, Mark. You point out, obviously this project wouldn't be happening without the initial support from Foundation Building Strength. And I think this is a great example of how foundational support can really help as an initial part for a bigger study. And the work not only with the CRISPR, but also with a different project that came out of the zebrafish model has led to the funding that Mark just mentioned from the Greenwald Foundation. We also have support from Muscular Dystrophy Canada for the CRISPR project. And then we have a really nice big NIH grant to cover a different therapeutic strategy for Nemaline that, again, started with support from Foundation Building Strength. I just want to mention people in my lab who've done the work. In particular, Nezrin Sabah has been the one leading the CRISPR-Cas9 project. We've had several excellent collaborators for this project. This is my larger team and some of the other funding sources. And just to give a visual of my happy lab, and we're very excited to hopefully really, I think this idea that Mark's mentioning of trying to accelerate research with a large amount in a with very tight deliverables in a shorter period of time is I think a great approach, and hopefully is we think that we're going to be able to really deliver on this. And the fact that we have this funding has now enabled me to have multiple people working on this project and to really approach it through all facets so we can get it moving forward.
I see quite a few empty pitchers there, Jim.
What's that? Yes, this was our holiday party, live pre-COVID holiday party.
Okay.
So I think we were anticipating COVID coming, so we did extra celebration.
Okay, so this is great. I just want to take another minute to give you a brief update on the foundation's happenings, and then we can go into questions for Dr. Dowling real quick. But I want to say that this year we have seen a almost fourfold uptick in our donation and granting dollars. And so if we would usually raise around $400,000 to $450,000, and with the Bikori grant, we just got the Chan Zuckerberg Initiative grant also. That's a $600,000 commitment over a 3-year span, so $200,000 per year. And so with those grants, in addition with a new board member named Sam Lipsick, who's giving us a whole new donor profile that did some great fundraising. Sharon Shimanovsky just had another $50,000 grant from another family that came into the foundation. And then we have the Machado family. They joined our board as well. And the Machados actually have committed $450,000 per year for the next 5 years. And they're also doing their own individual fundraising as well. And so we needed the money to get this momentum going even further and faster. And so we feel like there's more drive than ever right now to actually produce results. And I was telling this yesterday as we got together here in New York for the marathon. The 5 runners were there. I said, this is a marathon. But finally, for the first time, I feel like we can see the finish line in the distance. We're actually turning the corner, have some really positive momentum at the fundraising, really positive progress with the researchers. They're collaborative. They're not like keeping their secrets to themselves. They're really a very inspired and sharing group of the individuals that I've interacted with. So it's really great to see. All right, so I want to take a minute to turn it over to any questions. Anna, go ahead.
I don't really have a question, but I just want to say thank you so much for this presentation and for the job you are doing for all of us. Me and my daughter was the 2 persons on 2015 that had TPM and did all the testing. And I was so tired after that day. But I remember, I can't remember who it was, but I think it was one of the doctors from the Netherlands. He looked at me and he said, I can see in your eyes, you need to go to rest now. And I was tiring. And I remember I had aches in my legs for 2 days after I had been walking so much in stairs and on the floor for 10 meters due to, I usually sit in my wheelchair. But I am so thankful. And those 2 days with ache all over my body was so worth it. And I am so thankful for the work you're doing for me as an adult, but absolutely for my child and the rest of the children and their future. So I get chill when I see all and what you're doing. And I and the rest of Sweden are so thankful for your work, and also so thankful that you are able to do this for us today so we can listen to you. we had everything over here. I mean, we don't really have fundraising and things like that. Everything is funded by the state over here. So if someone wants to do a research on MN, the state has to prove it to get funds for it. So we are so thankful for being in the family over in America, as we call them, and for your work. So thank you for everything you do. And we are so thankful for listening to you today. Thank you so much.
Oh,
Thanks, Anna.
well, thanks so much, Anna.
Tobias, you're still on mute, sir.
Amateur. Yeah, thank you also for your work. We would like to know, because our daughter has the NEB gene, the problem with the NEB gene, and we're wondering, is there a timeline for when we can start some therapies, or excuse my bad English?
No. Yeah, so timelines are really always difficult with therapy development because there's things that are sometimes hard to predict in terms of what might be needed, and there's not just finding a potential target therapy, but also studies related to safety and toxicity and most effective dosing that needs to be done to really optimize things. So there's kind of two parts. There's the discovery part, but then the development part. And sometimes the development part can take quite a bit of time. But I think as what Mark is saying is that we're really hoping to use these innovative funding strategies to accelerate the development part. And we have this CRISPR project for the exon 55, but we also have another project that's looking at a way of therapy across the entire Nemaline gene that we think could potentially be effective regardless of the type of Nemaline mutation that you have. So those things are all underway. Unfortunately, it's hard for me to give a specific timeline because until I think there's still needs a little bit more work to know what's working exactly and what the pathway is going to look like to predict what that timeline could be.
Yeah, of course. Thank you.
Joanna, go ahead.
All right, but your timeline, Dr. Jim Dowling, is only for a year, and it's only going to be on the mouse models. It's not going to, you don't know if it will be expanded to bigger animal models or.
Yeah, so the goal at the end of the year is to have tested whether or not this is an effective strategy in an animal model, suitable to then consider for translation to patients. And the next steps will then be related to, well, what are the elements required from a safety perspective? What will we need to demonstrate before we could bring the therapy to patients? A little bit of that is a moving target as we get more knowledge about the safety of CRISPR-Cas9 as a general strategy. So the first clinical trials have just started really for any conditions. The first demonstration of the usefulness of CRISPR was done in patients with blood cell disorders. But in that case, they took the cells out of the patients, did the CRISPR-Cas9, and then put them back in. So, whether or not it's going to be effective in the whole body setting and whether it's safe, I think within the next year or two, we'll have a sense from other conditions what that looks like. And that will dictate a little bit what the safety requirements are pre-patient or pre-clinically.
Great question. Yeah, and it's great to see that there's models that have been successful in the past.
So
and
I think we'll be riding on the shoulders of others, which is fine because that part of thing is, it can often require a lot of, the fact that other groups are testing the safety of the Cas9 by itself, for example. We think there'll probably be very little safety concerns about the individual other parts of it. So we'll get a good sense for that from these other programs.
sorry, can I? So when you say the other programs, you mean the Duchenne community? Yeah.
Yeah.
Any others? Are you?
That's the one I'm most familiar with, because that's the neuromuscular disease where things are most progressing. And I think that's probably going to be the condition where we learn things first, because in general, as a rare disease, it's also the one where there's maybe the most active amount of research ongoing. And I know there's a couple pharmaceutical companies whose sole purpose is to develop CRISPR for Duchenne. So I think we'll get some insights from them. Whether there's other conditions as well, I don't know. My guess is probably, but those are likely to be the first programs to go into patients.
Okay, if there aren't any more questions.
Yes, I have
go
one.
ahead.
So, we're hoping, everyone here is hoping there will be a medicine for the NEB, ACTA1, all our gene mutations. Hopefully that day when there will be some kind of treatment or in a clinical phase, do you think it will be, since it's a rare disease, there's not so many patients to test on, do you think it will be a worldwide testing, or why I'm asking is because since we live in Sweden and you are in the US and or Canada, sorry, and I'm thinking that maybe I'm hoping it will spread to us as well.
Yeah, so I think, I would answer that in two ways. One is, I think it's very likely that any clinical trials will be done internationally across multiple sites, US, Canada, Europe, Australia, Japan. The network of clinical researchers. I mean, I think as Mark mentioned, one of the things that's a very positive is that we all work together very closely, very collaboratively. And I think the sense is recognizing that one needs all sites across the world in order to do a proper clinical trial. And also that by doing that, that enables participation across the world. The other thing I would say is it's important to understand that like the clinical trial piece is critical for the testing part. But if a therapy is effective, then there should be means of it being accessible across the world. And so, and that's a sort of second phase of a therapy development where clinicians and advocates are really key to spreading that word. If something is effective and it's been shown to work, is to get jurisdictions to pay attention to it. And and one of the good things in Europe right now is that a lot of that is occurring not just at the country level, but also in the whole continent level. And there's a lot of attention being paid to figuring out how to get rare disease therapeutics into all of the countries if possible.
And just to follow up too, there are other studies that we're funding that are looking at medications that might be helpful, to NEB and all different Nemaline myopathy patients. There's a study going on in Australia with Dr. Robert Bryson Richardson right now, and he's actually identified 5 different FDA-approved drugs that actually have shown the fish to be able to swim with a little bit more endurance, a little bit more speed, a little bit more power with these medication targets. And so they're actually doing some proof of concept work on the solution amounts and things like that to see whether those are therapeutic targets that could be palatable by humans. And so I think that's a great target. Then there's a lot of other small molecule work that's been initiated. There's a company called Myocardia that has an actin, improves the affinity of actin to grab the myosin binding site. And so that work is being done right now by Julian Ochala. And so we're really hopeful. We actually are starting an academic and industry working group. And so we have 4 different industry companies that have been asked to come to the table. And so that's hopefully going to start. We're going to have some invited researchers and clinicians join that working group as well. And there's a fellow who's was a higher up at Myocardia and Edgewise that are going to help lead the working group from the industry side. It's really hoping to get some big money pharma involved in our projects as well. Okay, I really want to.
Can I just say something?
Yeah.
I mean, a couple of years ago, you have to fill in here now, Johanna, since I'm not on the board due to we decided that only one parent each family can be on the board, and Axel is on the board. So we started a small organization, a family organization for NM in Sweden. And we will really be happy for every new information that you have that you can send to Johanna and I. Due to, once again, since everything over here is state funded, and since we are such a small community, even though some of our doctors sometimes go to Europe and have meetings, it will not spread all over. We still don't know how many who has NM in Sweden, and because we don't have that system. Wilma's doctor is one of the doctors now. They are trying to get to find out whereabouts in Sweden everybody are placed. But at the moment, we realize that the only way we can get the newest information, and we really saw that as an issue when we had this state-funded gathering for NM families in Gothenburg 2 weeks ago when we reached out to you, that the state kind of think, and the hospital and health organizations kind of think that they do have the latest update, but they don't. You have it. So we would be really happy if you can help us out with the latest information, so we can get more involved over here in Sweden, also help out with fundraising, and also get the pressure on our own country and our own doctors to be a part of all this success that we are doing.
Anna, thanks for that, because that's exactly what these working groups that Jim mentioned before and the one that we're developing as well are meant to do. It's meant to have the patients really influence the direction of research, the direction of targeted therapeutics. So I think it is underway. We have a lot more work to do. What I can do is say that if we wanted to have one representative attend our board meetings that we host, they're every other month, Thursday morning, the first Thursday of the month. We just had one last week. So it'll be again in probably January, the first Thursday in January, and it's at 9:30 in the morning Pacific time, 12:30 East Coast time. If you wanted to kind of assign a single representative to sort of, to jump into the Zoom like this, you'd be welcome to. So that would be, very easy. And then we could share any notes. They sometimes with the research studies, we have to wait to share bits of information. We receive a 6-month and 12-year report from the researchers. I'm going to say, if there's no more technical questions, I'm going to let Jim go on to his.
So thank you so much.
Have a great day.
Thanks so much.
You too. Thank you so much, Jim. Thank you.
Thank you so much.
So really, again, you're welcome to have somebody attend. We should be better about, sharing our notes. But I will have so Gus Japolsky is our medical director, and he summarizes in lay form the all of our reports so that we can post things. So it's usually pretty low, it's like kilobytes of material rather than this whole big download dump. So we'll try and do a better job of sharing more of that information in a larger format, because we wanted to influence your state. We wanted to influence your medical practitioners.
Yes, thank you. And I was thinking,
the runners are. We have one of the runners passed two of the runners passed the halfway point That's my
Oh,
friend.
that's great.
My friend Mark Johnson, right there. MJ is running a pretty good pace. Felipe is really tearing it up, and then Misty started later. She's just past mile 8, and
okay, yeah, she's running for Greta, right?
Yeah.
That's great. Wow. Can you hang on? Can I just say some Swedish words fast with Johanna?
Ja.
Johanna? Ja, jag tänker så här, och ni alla andra, för Johanna och jag har pratat om det här tidigare. Jag kanske den som har lättast att koppla om, för jag kan sova på morgonen med mitt arbetsschema. Så jag tänker att jag kan erbjuda mig att vara med på det mötet, första mötet, och sedan kan ni i FN ta ett beslut om vem som ska vara den som blir uppkopplad till de här mötena.
Absolut. Vi får prata om det på styrelsen. Absolut.
Ja, men så vi kan säga, jag tänker att om vi säger att någon av oss kör, men jag kan ta det först och så får ni ta det som ett styrelsebeslut sen.
Låter jättebra nu. Vi kör på det.
Yeah. I'm back. We have decided a little bit over here now.
Sorry, what was that?
Yeah. We will really be happy if I could be the one who's included on the first meeting, and then we'll take it to our board and see who's going to attend the rest of the meeting. So I think you have my email, and you can just add me for the next meeting.
Sounds good.
Great, thank you so much. I'm the one that's working mostly from my home office, so it doesn't matter what time it is. So I can sleep when Wilma is at school.
Joanna, do you have one more thing?
Yeah. So you were saying we want to contribute. I'm thinking in all other countries, because you're doing so much in the States, and that was actually my question. What can we do to contribute? What's the easiest way to get involved with this? You were saying was called the Treat NMD Congenital Myopathy Task Force, and I'm thinking in contributing with AFBS as well. What can we do?
Yeah, thank you. So as part of our PCORI grant, I mentioned that we have to do 10 webinars, and the webinars, we estimated that we would have 75 people attend. So our goal is to get about 50 different family members to attend those meetings, and the others could be, board members of AFBS, and then targeted researchers. And we're still plotting out what each of those different webinars will be from a content perspective. We've thought about, just across those 10 different meetings to do the journey that someone with Nemaline myopathy goes through. So whether the first one should be on, what the mom experiences in pregnancy, what the genetic confirmation process is like. So have the early geneticists involved in those meetings. So that's still in debate. We have some plan sketched out for those meetings, but I'll just share whatever I can content-wise with you. You're welcome to see the full confirmation agreement that we have is public. The PCORI grant that we have is it's a public grant, so you're more than welcome to see what, and we would love to hear from you if you have ideas about how we can improve on what we deliver. From a fundraising perspective, we also as a foundation just switched organizations that run our contribution mechanisms. So the actually international donations are a lot easier. Well, we were using a group called Network for Good before that blocked a lot of international credit cards and things like that. But now a new organization called Classy is reduced and changed the security so that it's got a protection for European and South American contributions as well. So there might be a great way to try and start a fundraising operation within Sweden. That would be great. And we see things as simple as a garage sale. You sell some products that you've moved on from, and then you just when people start bartering with you, you can say, well, this is for charity. Like we're And so I think that just being able to do that would be an opportunity for, folks. I don't know if you guys know this, but there's a organization called Alex's Lemonade, which is a kids cancer organization. And they basically said, set up a lemonade stand outside your driveway and send the money to them. And they raise about 25 million a year from
Wow.
a lot of the majority of it is from these small group organizations. Every child that's diagnosed with cancer has the opportunity to sort of do these emotional fundraising events, and they end up charging, $20 for a glass of lemonade. And, anyway, it's a great way to kind of get things initiated on a small scale. And then when you see how that momentum can build, it can really take off.
Great.
Well, let's Joanna, I'm pretty responsive by email, as you probably noticed. So let's just stay in communication, and I'll try and be as best I can. If I happen to fall off the radar, just please ping me again. And then we'll just keep wrapping back, because I'd really love to keep this conversation going.
Great.
Great. Thank you, Mark.
Yeah.
Very. Thank you so much for doing this for us. It's been listening to Jim and listening to you. It's been really, really interesting, and it's seeing where the science is right now. It's amazing, and we're just hoping that it's evolved so much. It's evolved so much like the last couple of years, and we're just hoping it will be a bright future for it. Really, thank you.
Yeah, you give us hope, Mark.
Yes, you do.
Well, hopefully we deliver on more than just that.
Yes, yeah. Yeah.
All right. Well,
Great.
thank you, everybody. Really appreciate your time today, and look forward to future contributions. You all can help us, in steps forward. Okay.
Thank you. Take care now.
Bye now.
Bye. Bye.
Bye.
Bye.